Sorry, there's somebody at my door. Okay, this is part 2, I guess. So I left off here. So we said this protein channel will naturally diffuse hydrogens from high to low concentration if it's open. This channel is always open. So it's kind of constantly having this flow of hydrogens out. But then as long as there's light present, we have this constant push or pumping of hydrogen into our plant. So as long as we can maintain this gradient from high to low, then we can kind of constantly have hydrogens moving out. If we reach, well, it's not isotonic, but if we reach the point, well, yeah, isotonic, where the concentration of hydrogen in and outside are the same, then you're just going to have some hydrogens will move out, not many, right? And then when light is present, we'll kind of pump more in, and as we pump it in, we'll kind of move it out. So you have this kind of constant movement of hydrogen coming in, hydrogen going out. And the purpose is that this ATP synthase is actually shaped like a rotor. And what a rotor does is it can spin, or it rotates. And when the rotor rotates, it creates kinetic energy, which can then be stored as chemical energy. So we're going to rotate our rotor-shaped enzyme. So this protein will literally rotate, and when it rotates, the energy of that rotation is going to be stored in ATP molecules. So here's how we do it. These hydrogens that have flowed in, they're going to pumped in, they're going to start flowing out. And the only way for them to get out from high to low is through ATP synthase. Each time a hydrogen moves through the ATP synthase, it causes the ATP synthase to rotate. That rotational movement, oh, my cat is annoying. That rotational movement is it creates just enough energy to take an ADP plus an inorganic phosphate and generate ATP. So the energy from the rotor, this rotational kinetic movement, is now stored as chemical energy in our ATP. ADP to ATP is an endergonic reaction, so it got its activation energy from the movement or the kinetic energy of this ATP synthase. The movement of this ATP synthase occurs as protons flow through the protein and cause it to change shape. So it's kind of changing shape in such a way that it's rotating, generating just enough energy to make some ATP. So the more hydrogens we can pump into the lumen, the more hydrogens will move out through ATP synthase, because that's their only way to diffuse, the more ATP will generate. Okay, so now we have created some high energy molecules. So these two things that we drew crossing the line, those are our two products of the light reactions, right? And our reactant, we had H2O, we had NADP+, we had ADP and inorganic phosphate. And now we have these high energy molecules. So what these molecules do is they're used to run the Calvin cycle. So we're going to draw the Calvin cycle as a circle. So let's kind of just make a note. These two are going to power the Calvin cycle. And we'll kind of draw them coming into the Calvin cycle at specific times. So if you remember, for our equation, we now need to create this really large molecule of glucose. So essentially what we've done is we've taken all the energy from the water, and we've created oxygen, right? So there's our product of oxygen right here when we split water. So now we need to figure out how are we now going to take the carbon from carbon dioxide and put that carbon into these glucose molecules using the energy that we got from sun and the electrons that we pulled from water. So the Calvin cycle, it's a cycle, meaning the molecule that you start with is the molecule that you end with. So we draw it as a circle. Okay, so the Calvin cycle starts with a molecule. It's called RUBP. And then at the end of the Calvin cycle, we're going to recreate RUBP. So it's essentially a 3-phase cycle. She is upset with me. The first phase is called carbon fixation. The second phase is called reduction. And the last phase is called regeneration. So this is where our carbon dioxides will come in. So if you remember our reaction equation, 6 waters plus 6 carbon dioxides will yield a glucose and 6 oxygens. So those 6 waters and the 6 oxygens, so these waters came in the light reaction, we created our water in the light reaction. So these last 2 reactants and products will be for our Calvin cycle. So the carbons are going to enter the Calvin cycle one at a time. So we're going to add in our carbon dioxide to our first stage right here. Okay. Eventually we are going to add 6, just like our overall reaction states. The issue is we can only add 1 carbon dioxide at a time because on the enzyme that's going to do carbon fixation, there's only 1 active site. So we're going to add our carbon dioxides to cycle 1 at a time. Meaning it takes 6 Calvin cycles, so this will go 6 times, to make 1 glucose molecule. Okay, so you're going to kind of be constantly outputting these intermediate molecules until you add enough carbon in so that you can take out a 6 carbon molecule. So the carbon fixation phase is we are taking this carbon from carbon dioxide and we're fixing it to or binding it to the molecule, the first molecule of the Calvin cycle. The first molecule is called RUBP, also known as ribulose bisphosphate. So that's our first molecule. So we need to get RUBP and carbon dioxide to bind with each other. The enzyme that causes those 2 things to bind with each other is called rubisco. It sounds like Crisco. Rubisco. It's an acronym. I can't remember what it means, to be honest. So rubisco is an enzyme. And this enzyme is going to bind RUBP with CO2, and it's going to create a product, which is both of those things together. That's the carbon fixation stage. It's going to take energy to do that. So we're going to use this ATP here to run the carbon fixation cycle. So we're going to take ATP that we got from our light reaction. We're going to add it into the carbon fixation stage. We're going to end up with ADP and our inorganic little phosphates. So that uses our ATP from our light reaction, and that will power the creation of this molecule because that is powering an endergonic reaction. Next, we're going to reduce this molecule that was created. Okay, so we took RUBP, we bound it with CO2, so we carbon fixed it. We had energy added to run that chemical reaction. Now we're going to add electrons to that molecule, and those high energy electrons are going to come from our NADPH, which is shuttling those electrons. So we're going to take our NADPH, draw your little electrons, what did I write? NADPH. Those electrons are right here, so those high energy electrons are there, but they're going to get snagged, and you're going to end up with NADP plus, and then some free hydrogen ions. Okay. So the electrons that were here have now been placed here. Okay, so these high energy electrons are now bound to the molecule that is kind of cycling through here. So now the molecule here has been reduced, it gained electrons. Our NADPH has been oxidized, it lost its electrons, and now we have NADP plus and some hydrogen ions. So now the molecule that's going to be moving through is this high energy molecule. It got energy from ATP, and then it also got high energy electrons from NADPH. At the end of the reduction stage, we're going to release a molecule. So for every 3 carbon dioxides that enter, so we'll go once around, twice around, 3 times around, you're going to release a 3 carbon molecule, okay? So you're going to release 1G3P. So a G3, the 3 stands for the number of carbons in this molecule. So we have 3 carbons. So you release 1 glyceraldehyde, 3 phosphate from this reaction. Okay. And then the next stage will regenerate RUBP. Then one at a time, you'll enter the 3 CO2s. You'll release 1 more G3P. So let's kind of write down what happens over here. So once you have 2 G3P molecules, you can only get 2 G3P, glyceraldehyde 3 phosphates can be released only if you add all 6 of your CO2s. Because you can only take carbon out if you've added that carbon in. If I start with ribulose bisphosphate and I need to end with ribulose bisphosphate, I can't just take carbons out because this is a carbon-based molecule because I won't end with that. So the only way for there to be an excess of 6 carbons, 3 times 2, 6 carbons, is for me to have an input of 6 carbons. So the 6 carbons that we enter through carbon dioxide are the 6 carbons that exit as glyceraldehyde 3 phosphate. Once I have 2 G3Ps, you can actually, these will bind together if they get to the correct enzyme, and that will create for you 1 glucose molecule. Because you need 6 carbons for glucose, C6H12O6. We have 6 carbons right here, so these are kind of like the precursor to our glucose molecules. Okay, so this is our product. We have made our glucose. Wonderful. Now, last thing to kind of look at, the rest of our products here. So we had ATP go in, and we have these products, ADP and phosphate. Those are actually just going to go back to the light reaction and be used right here. So we're kind of constantly cycling this ATP, ADP matter and just regenerating it. Same thing with our NADP plus and our hydrogen, right? So that's just hydrogen ions now are in the stroma, just like they were over here. They might get pumped back into the thylakoid, diffused back out. Our NADP plus is just going to move back over here to its enzyme where it has its active site, and it's just going to get reduced again and then keep shuttling electrons back and forth. So everything kind of gets moved back and forth between the two reactions. So let's kind of summarize what we've done here. So for our light reaction, where it takes place across the thylakoid membrane. So it's accessing both the lumen and the stroma, so it has a differential concentration gradient. So you can kind of conclude that the more folds we have in our granum, the more thylakoids we have, the more electron transport chains we can have, right? So like this single granum can have multiple electron transport chains, multiple ATP synthases. You can be creating like hundreds of these molecules at a time to run hundreds of different Calvin cycles. So for the light reaction, what we used from our equation, zoom out a little, what we used from our equation up here, we utilized our 6H2O as our reactants, and then we generated our 6 water molecules. So they were split water molecules. So you actually get oxygen not only from splitting this water, you get oxygen from the Calvin cycle as well. But the bulk of your oxygen released is going to come from the splitting of water. And then you also have, there's an input of oxygen over here. We just don't write it because the overall reaction has an output of oxygen. So that's kind of like what we've used. We split water and then we release oxygen from that. And then we also created our high energy molecules. We've created ATP and our high energy electron shuttle NADPH. So these are kind of like all the things we've generated. All of this will kind of transfer, so our ATP and NADH transfer to our Calvin cycle. It occurs in the stroma. Okay, we do have an input of our carbon dioxides, and then we're going to eventually release C6H12O6 in the form of 2 G3Ps, right? So essentially this is 2 glyceraldehyde 3 phosphate molecules, and they'll bind up to make a glucose. You've also created ADP and an inorganic phosphate. We've also created NADP pluses, that's a plus, and some hydrogen ions. So those are kind of things that were released back into the stroma, but they can be used back here in the light reaction. So you can kind of see we're kind of constantly sharing molecules between our two processes here. But our end goal was to get this. And so once we have this molecule, the plants can use it to build more cell walls, so they can do growth and development, make more plant, they can reproduce, or they can ship the glucose off to the mitochondria, and then the mitochondria will run cellular respiration, which is what we're gonna go over next. Okay, I'm probably gonna post a couple Bozeman videos just because he kind of goes over some specialty plants that I wanted you to review. And then I'm going to put practice FRQs and stuff up for you guys. All right, I'll talk to you later. Bye.